Hello, everybody. Welcome to this webinar, the first of a series of webinars in May 2025 that are dedicated to FSHD. And this webinar is organized by EURO-NMD in collaboration with the European Reference Network for Rare Neurological Disorders and the European Academy of Neurology. And we have as first speaker Professor Emiliano Giardina, that is Associate Professor of Medical Genetics in Tor Vergata University of Rome since 2015, where he holds directorial positions at the Santa Lucia Foundation in Rome, overseeing both the lab for diagnosis and medical genomics and the genomics medicine lab that is connected to the Italian Union against muscular dystrophy. He also serves as director of the Forensic Genetics Laboratory at the same spot, where he collaborates with Police Forensic Science Unit of the Anti-Crime Central Direction. Professor Giardina's research primarily explores the molecular characterization of Mendelian and complex diseases, particularly focusing on ocular, dermatological, neurodegenerative, and neuromuscular disorders. His efforts have significantly advanced the genetic understanding of these disease mechanisms and has helped develop new diagnostic and prognostic protocols and enhance pharmacogenetic responses to treatment. So he will be sharing his knowledge about the genetic side of FSHD, and we are all eager, I'm sure, to hear to him. So I'll shut up and I'll pass on the floor to Emiliano. Thank you very much for accepting this invitation. The floor is yours.
Thank you, Antonio, for your kind presentation. I hope to be at the level of that. So good afternoon, everyone, and thank you for joining this first webinar on facioscapulohumeral dystrophy, and in particular on the genetic diagnosis of FSHD. It's a pleasure for me, it's a real pleasure being here and sharing with you some of my thoughts and the recent advances in the genetic diagnosis of this really complex disorder. But first of all, I feel to thank the organizers, but in particular Nicole Verman, for all her efforts in coordinating a fascinating but also sometimes challenging network of scientists, in particular for the revision of the genetic guidelines for the diagnosis of facioscapulohumeral dystrophy. Okay, so in this slide, I want just to summarize what will be the content of my talk. So a presentation of genetics of facioscapulohumeral dystrophy and the distinction between the form 1 and 2, the role of the DAX4 gene that is central for the pathogenesis of the disease, the genetics and epigenetics regulation of this gene. I would like to give you just an update of the new guidelines I mentioned before, some piece of information about pre-implantation genetic test, as well as prenatal diagnosis, genetic counseling. I would like to stress the importance of genetic counseling, not only for patients, but especially also for healthcare providers. And also, I would like, if we will have time, one or two practical examples of that. So, facioscapulohumeral dystrophy is part of a group of muscle dystrophies, differing in terms of the clinical presentation, the genetic causes, and the affected muscle groups. And as the acronym can suggest us, it follows the temporal clinical course of the disease. The disease typically affects the facial, scapular, and humeral muscles first, and then we have a later progression to trunk and lower limb involvement. So it is important, in my opinion, just in these first slides, to outline that while the clinical patterns can guide the diagnosis, every genetic disease has always a clinical diagnosis, genetic confirmation is essential, given the overlap and the variability that we can observe among all these muscle dystrophies. So the importance of the genetic diagnosis is not only for the patients when he is positive, but also when it is negative. So we can look for a differential diagnosis. So the disease was firstly described in 1885 by two French neurologists. They observed for the first time a specific familial pattern of muscle weakness involving the face and the shoulder girdle. So in this slide, I would like to just summarize some clinical features. We will go through this in details after. So the first important issue to consider is that this disease is characterized by a huge clinical variability. It can present in childhood, adolescence, or adulthood, and its expression can be asymmetric in many cases. Although it should follow an autosomal dominant inheritance pattern, the penetrance is incomplete many times, and the expression is highly variable, even within the same family. So we can have within the same family different patients with different phenotypes, but with harboring the same genetic condition. So the disease is autosomal, and so the disease affects males and females, but some studies have suggested higher penetrance in male. So here I want to summarize these main features of facioscapulohumeral dystrophy, including the epidemiology, the pattern of inheritance, the molecular diagnosis, the standard of care, the clinical features. So it is the third most common myopathy in humans, affecting 1 of 10,000 individuals. And the pattern of inheritance is autosomal dominant. As I say, the molecular diagnosis, standards of care, or clinical features will be covered later in this presentation. So I would like just for the patients, I would like just to give some piece of information about the autosomal dominant pattern. Autosomal means that the gene in question is located in one of the non-sex chromosomes. Dominant means that a single copy of a mutated gene from one father, the mother of the father, is enough, a single allele is enough to cause the genetic disorder. At the moment, more than 1,500 dominant diseases has been described in humans, and it is and there's an estimation of that on average 1% of live born can present an autosomal dominant disease. So what about the genetic risk? Each gamete from an individual with a dominant trait or disorder will contain either the normal allele or the mutant allele. So the risk to transmit the affected allele is 50% for in the case of facioscapulohumeral dystrophy. We have mentioned the reduced penetrance. The penetrance is a concept a little bit challenging for not specialized people, but the penetrance in easy words can be defined as the proportion of individuals carrying a particular variant of allele of a gene that also express an associated trait. So in some individuals, they can harbor the genetic mutation without expressing the disease. These are called reduced penetrance or skipping a generation, in other words. The result, what is the reason of that? Probably the genomic environment of the individual, the effect of other genes, as well as the effect of environmental factors. This is exactly what we observe in facioscapulohumeral dystrophy, where we can observe patients severely affected and patients practically asymptomatic, sometimes with the same mutations. So last, I would like just to spend some words about the variable expressivity. That is it is a clinical feature typical in autosomal dominant disorder that can show a huge variability in the expressivity of the disease, even in the same family. So this image shows us a typical patient affected by facioscapulohumeral dystrophy with a prominent scapular wing and the lumbar hyperlordosis. These specific clinical signs, especially when we observe a positive family history, should raise a strong suspect of facioscapulohumeral dystrophy and request the genetic testing. However, we have to say that atypical cases exist, and so there's a need not only of the molecular diagnosis, but also a continued cooperation between geneticists and clinicians. And this cooperation can find her place during the genetic counseling. So here I would like to show a family from our experience in Italy. One of the most intriguing aspects in the facioscapulohumeral dystrophy is its intrafamilial variability. Here we can see a beautiful example. One sibling presents with a severe phenotype, but the other one was practically asymptomatic at the age of 42. So the clinical heterogeneity is attributed, it has been attributed for many times to unknown genetic and epigenetic factor, genomic environment, and so on. However, this was exactly the starting point of my experience in studying the facioscapulohumeral dystrophy genetics. But at the moment, I want to give you some words about that, on the fact that a large majority of this variability now can be explained by refined molecular tools, especially with whole exome sequencing, methylation analysis, or second-level analysis. So at the moment, we are in a better position than some years ago. It's important also to give this perspective, in my opinion, to patients that research go on. We all know that there are two forms of facioscapulohumeral dystrophy. The form 1 affecting the large majority of patients, probably 95% of patients, and the form 2 affecting only the 5% of patients. They share exactly the same clinical features and both are caused by inappropriate expression of the DAX4 gene in the skeletal muscle. So for the patients, probably this distinction can be not so relevant, but for us as clinicians and researchers, it is crucial for a good diagnosis, for the genetic counseling, for the clinical trial, and also to give the informed information to the patients. So just during these first slides, it is clear that the genetic mechanism underlying the facioscapulohumeral dystrophy is really complicated. And it is so complicated that we could easily write an entire textbook of genetics only dedicated to this single genetic disorder. The interplay between repetitive element in a human genome, the epigenetics, the genetic regulation of the gene expression, the haplotypes, the gene regulation makes facioscapulohumeral dystrophy, in my opinion, one of the more extraordinary models for understanding the gene expression, the regulation of the gene expression, and also the chromatin biology. So I would like to spend just briefly some words on the DAX4 gene that is essential for the facioscapulohumeral dystrophy pathogenesis. The DAX4 is a double homeobox transcription factor that is normally expressed only in the early embryonic development, especially in the cleavage stage embryo. It plays a role in the zygotic genome activation, regulating a network of genes involving the totipotency of cells. In somatic tissues, including muscle cells, the DAX4 is epigenetically silent, so it is repressed. And unfortunately, in facioscapulohumeral dystrophy patients, this repression fails due to the contraction of a specific array that we call D4 zeta 4, or due to the mutations in chromatin modifiers the DAX4 locus can become epigenetically de-repressed. As a result, the DAX4 is expressed in the normal cells, and it can cause the disease. The level of the expression can be really low, because this even low level expression and sporadic expression of DAX4 is sufficient to activate a toxic transcriptional program leading to the disease. It is important to spend some words about that, because it enables us it allows us the impossibility to detect the disease to diagnose the disease, detecting the mRNA or the expression of the genes. The gene is expressed at too much low level to be detectable by our current analysis. And so we have to perform the genetic diagnosis in a different way. So this is just a schematic representation for patients. When the DAX4 is silenced, the patient, we don't have the disease. When DAX4 is expressed, we have the disease. So here I would like just to summarize some of the most important facts in the facioscapulohumeral dystrophy type 1 and type 2. It is not easy, but it's important for me just to have clear in mind some aspects. So the facioscapulohumeral dystrophy is associated with the deletion of a chromosomal tandem repeat called D4-zeta4 near to the distal end of the chromosome 4 at the position 4q35 location. This region is a polymorphic variable number tandem region we call VNTR, consisting of 3.3 kilobase units. Each unit encodes for a single DAX4 gene. So in healthy individuals, we have a number of repetition, a number of units ranging from 11 to more than 100, 150. In individuals, in patients affected by facioscapulohumeral dystrophy, this number of repetitions is reduced from 1 to 10. And this contraction array causes the hypomethylation and relaxation of the chromatin, which activates the expression of the toxic DAX4 gene. So the facioscapulohumeral dystrophy, at the end of the day, both the type 1 that we are talking about, and the type 2 is caused by an ectopic expression of the germline transcription factor DAX4 in muscle cells. However, this picture is can be even more complicated by the fact that the contraction of the DAX4 of the D4-zeta4 array on chromosome 4 by itself is not sufficient to cause the disease. Proximal to the D4-zeta4 region, toward to the distal end of the chromosome, there's a polyadenylation sign that is required, is a region required for the DAX4 gene, required for its stable expression. And so without this part, the gene cannot be expressed. We cannot find the gene in the cell. So to complicate even more is the fact that the chromosome 4 comes in two different alleles. So we can have two different haplotypes, the 4QA and the 4QB. And the 4QA is, sorry, I'm looking for my pointer. Okay. The 4QA is a permissive allele, and the 4QB not permissive allele. And therefore, only the 4QA variant in combination with the contracted array region is associated with the disease, because we have the hypomethylation, the expression, and the stabilization of the mRNA. On the other hand, individual with a contracted array, but with a non-permissive allele, will be completely unaffected. So during the genetic diagnosis, we have not only to understand if the patient harbors or not a contracted allele, but we have to look also to this part, so this haplotype. And so, however, interestingly, we have to say that this array is not only present only on chromosome 4, it is also present also on chromosome 10. However, this contraction is not pathogenetic because due to the absence of the haplotype of this polyadenylation signal that I mentioned you before. So I mentioned you before that we have two forms of facioscapulohumeral dystrophy. The first one is caused by the contraction of the allele. The second one is caused by accounting for the 95% of patients. The second one is caused by some mutations in gene encoding for the epigenetic repressor of the DAX4 locus, and is associated with this mutation, but also when the repeat array falls within the borderline range, typically 11 to 20 in the literature, but in our experience also we can have this kind of mutation also in patients typically classified as facioscapulohumeral dystrophy type 1. So I would like just to give you just brief some notes about the facioscapulohumeral dystrophy genes. The most important one is the SMID1. It plays a key role in epigenetic silencing of by regulating the chromatin architecture. It promotes heterochromatin formation, so it is a repressor. And it plays a key role in the chromosome X inactivation. So the DNA methyltransferase 3 beta encode for a methyltransferase that function for the de novo methylation. It is essential for the establishment of the DNA methylation during the development. And last, just some words for the ligand-dependent nuclear receptor interacting factor 1 that is involved in dosal compensation, once again, in the inactivation of the dextro chromosome and co-localized with chromosome in the telomeric region. Together with SMID1 is involved in chromosome X inactivation in females. So it is important to outline that this epigenetic regulation, so that the repression of the DAX4 gene is the common denominator between the form 1 and the form 2. So both the forms converge on a state of D4-zeta4 hypomethylation, leading to the DAX4 expression in skeletal muscle. It's important to learn that because this shared epigenetic signature is not only for us a key pathogenetic mechanism, a specific features, but can become also a reliable diagnostic marker. So this picture, form 1, form 2, the repression, haplotype, contraction, mutation, is even more complicated when we consider that the form 1 and the form 2 are not mutually exclusive categories. So recent evidence suggests that this form represents two ends of a molecular continuum rather than two separate diseases. And so importantly, this kind of mechanism can occur in the same patients. So we can have some specific mutation in the modifier genes, also in patients with a number of units from 1 to 7. It is less frequent, but it happens. And in the large majority of cases, we will have patients with a number of repetitions from 8 to 10, so in the borderline range, but still pathogenetic, together with this mutation. So it is important to outline that the facioscapulohumeral dystrophy is not a single disease, but it is a huge number of different diseases on the basis of the combination of different genetic factors and epigenetic factors. So given this molecular complexity, the overlapping feature between the facioscapular humeral dystrophy 1, 2, the presence of intermediate case, the presence of atypical cases, it is clear that no single test, no single approach, no single specialist can be sufficient, can be enough to reach a confident diagnosis. Instead, we are looking for a multidisciplinary, a multimodal, approach, combining precise molecular genetics, including repeat sizing, haplotype analysis of the distal length for QA, for QB, the methylation profiles, a detailed clinical evaluation by expert neurologists. So a detailed, a standardized clinical evaluation. I like to mention the work that Giulia Ricci has done so far in Italy, but not only, to try to standardize the the clinical presentation of patients affected by facioscapulohumeral dystrophy to promote the genetic correlation between genotype and phenotype. We have also to cope with instrumental assessments such as MRI and electromyography. The experience of polyclinical gemelli in the analysis of MRI is really extraordinary. And also, we have to combining all of these things with a comprehensive genetic counseling to interpret the findings in the context of personal and family history. So this is the starting point. So now I would like just to give you some information about the genetic diagnosis of facioscapulohumeral dystrophy over the time. And so in this slide, you can see the most important scientific achievements that have made possible the current diagnosis of the disease. And so at the moment, we are experiencing some advanced technologies, such as molecular combing, the optical genome mapping, methylation profiling, that in combination with the gold standard, that it is the old technique, Southern blotting, can ensure a good diagnosis. So I would like, before to dive into the clinical aspect and the technical aspect of the diagnosis of facioscapulohumeral dystrophy, in my opinion, it's important to outline a paradox that makes, for my position, this disease truly unique in the field of medical genetics. Despite the advances in genetic diagnosis and genetic sequencer that we have for all the other genetic disorders in my laboratory, we are going to change our technologies every 2 or 3 years. But despite all these advances, the gold standard of facioscapulohumeral dystrophy still remain the old technologies, such as the pulse field, gel electrophoresis, and Southern blotting. This is due to the fact that the pathogenic mechanism of facioscapulohumeral dystrophy is not a point mutation, but a complex rearrangement with some homologies with other chromosomes that can make it invisible to the most next generation sequencing platform. And this type of mutation requires high molecular weight DNA, high quality and high quantity of the DNA. So it is important to outline that, because as a result, the facioscapulohumeral dystrophy diagnosis has remained technically demanding and highly specialized, limiting its implementation to a small number of centers worldwide. This was the starting point of our revision of the international guideline, to try to expand as possible the number of centers able to perform the genetic diagnosis of the disease. So there's no need for in this session just to go through the materials and methods of the diagnosis. It is only to know that the diagnosis process begins with the collection of peripheral blood. After that, we have a double an extraction of DNA, of high quality DNA, a double digestion, a double digestion, enabling us to discriminate between 4 and 10 chromosomes, and to enabling us to understand if the contraction is present or not. After that, we have the pulse field of the Southern blot, and we can check the analysis in a very old-fashioned way. And so this is the threshold, and if the bands will be at the end of the below the threshold, the patient is positive. If the band, if the contraction is above the threshold, the patient will be negative. So although the core diagnostic methods of facioscapulohumeral dystrophy is the Southern blotting, it is quite old technique. In my opinion, it would be misleading to say that technologically progress has not impacted this field. In fact, as I told you before, the recent advances in the genome analysis technologies contributed decisively to our understanding of facioscapulohumeral dystrophy, in particular in cases, atypical cases, borderline cases, in all those cases that some years ago could be defined not perfectly explainable. So I would like to mention one of these most important instruments. It is the optical genome mapping. It is one, in my opinion, best example of the coming future of the diagnostics. It enables the detection of all classes of chromosome aberration, provides 10,000 higher resolution respect to normal karyotyping, and can detect a huge number of structural variants that are or that can be missed during the evolution of the performing of current technologies. It is important because optical genome mapping is, in my opinion, a little revolution, not only in the field of facioscapulohumeral dystrophy, but also for all the other genetic conditions. So this is just an example that the validation of this machine is already done. So, as I told you before, this kind of machine, together with the extent of the research, demonstrated that a small percentage of facioscapulohumeral dystrophy cases, about 5%, are due to complex difficulty to type default zeta-4 rearrangements. These include unusual genomic events such as duplication, in cis duplication, and deletion. So what about the extended deletion? One patient, more than 1% of patients can be can be a carrier for this extended deletion. Is one of the more common atypical rearrangements. In this individual, the default zeta-4 repeat is partially deleted, but also the sequences immediately proximal to the repeat is lost. This causes a false negative because the failure of the hybridization of the probe that we use to understand if the contraction is present or not. And so, in this case, the diagnosis can be performed using molecular combing or a single optical mapping. Another cause of false negative result is the in-cis duplication, where the proximal default zeta-4 array in this duplicated allele is of normal size, as you can see in this cartoon, but the distal end is on contracted array. They are not distinguishable during the Southern blot, and therefore we can have a false negative result when the patient harbor a contracted allele in the presence of a 4QA haplotype, leading to the expression of the genes and the expression of the disease. So in our research, we are systematically applying the DNA methylation, the Southern blotting, and the whole exome sequencing to all patients with clinical suspicion of facioscapulohumeral dystrophy. And I would like to thank the Associazione Italiana di Immunologia for all her support in making it possible. And by combining these two layers of analysis, we were able to detect an higher than expected number of patients carrying pathogenic or likely pathogenic variants in genes involving chromatin regulation, such as the classical gene for the FOM2, but also others. And so it is important to say that research progresses every day. So every single day we have new papers, every single day we have new achievements. And so it is important to give this perspective to our patients, because we are working hard for all patients affected by facioscapulohumeral dystrophy. And so our diagnostic strategy is based on a simple but powerful, in my opinion, biological assumption. So we know that facioscapulohumeral dystrophy develops when the DAX4 genes is expressed. And we know that the DAX4 gene is expressed only when there's the hypomethylation of the region. The methylation is the brake, the hypomethylation is the accelerator. And therefore, if methylation is present, the levels are preserved, the DAX4 remains silent, the disease is not present. If methylation is lost, the gene becomes accessible, and we can detect the mRNA, and the disease can be present. Based on this simple assumption, we developed strategies using the DNA methylation analysis at first-line screening tool in all patients with a clinical suspicion of facioscapulohumeral dystrophy. It's important to say that it is crucial to avoid a huge amount of non-necessary testing. So patients that are negative for the methylation is unlikely to be a patient of facioscapulohumeral dystrophy. The increasing complexity of the facioscapulohumeral dystrophy genetics and the introduction of new technologies have led to the need for an update of the diagnostic guidelines. It was the work I mentioned before, and it was a very, very challenging work. We get an international consensus about that. And so the international guidelines, in our opinion, this is the starting point of our action. They should not only recommend the most advanced and expensive genetic characterization, but also account for the average capability of laboratories. This goal of our effort was to get a balance between the best available technologies, in many cases also expensive, but also identify a feasible common denominator, ensuring that all centers can be compliant with the guidelines. And for this reason, we identify action as essential and recommended. So these updated version of the guidelines were developed through a global collaborative effort coordinated by Nicole Vormans, Richard Lemmers, and myself, and involving experts from Europe, North America, Asia, South America, Australia, and so on. The goal was to harmonize practices and ensure the flexibility. So it is important to outline that we decided to analyze only 3 categories, to subdivide the patients into 3 categories: typical and atypical facioscapulohumeral dystrophy with a severity moderate to severe, typical with mild phenotype, atypical with mild phenotype. So it's important, before to dive into the details, in my opinion, it's important to emphasize a principle that is at the basis of this evolution of the guidelines. The diagnostic workflow now begins with the clinical evaluation. Rather than starting with the genetic testing alone, this updated approach recognizes the fact that the clinical phenotype is the driver, helping to define the level of suspects and driving the choice of the subsequent molecular investigation that can have the first level, second level, third level. So starting from this point, the workflow of the structure of the guidelines follows a tiled structure where the level of genetic and epigenetic analysis is adjusted based on how well the clinical findings correlate with the molecular data. In case where there's a strong genotype-phenotype correlation, a minimal genetic work can be sufficient to confirm the diagnosis. In contrast, when we have a suboptimal genotype-phenotype correlation in cases that are atypical or borderline, additional testing can be necessary, for example, methylation analysis, sequencing of modified genes, and so on. So I would like just to briefly to show these guidelines. So, for example, I show you the case of an atypical FSHD with a good severity. So, in this case, if the repeat array contains from 1 to 7 repetitions, there's no need to do something else. When we have from 8 to 10 units, the situation is less clear because we expected a less severe phenotype. So if the methylation and sequencing are negative, we can still remain on facioscapulohumeral dystrophy 1. If we have the presence of methylation suggesting the form 2 or the presence of the mutation in the modifier gene, we can diagnose as a form 2. What about the more than 10 repetition? More than 10 repetition, we have the situation is unlikely. The facioscapulohumeral dystrophy is unlikely. However, and once again, if we can check for the deletion and duplication, we can still remain on facioscapulohumeral dystrophy type 1 because of false negative. But if we have the methylation suggested the form 2 or the methylation or the sequencing of the genes, we can remain on the form 2. So I would like to skip this to go to dive. So the same structure, I'm sorry, is followed for the typical for facioscapulohumeral dystrophy with the severity mild and atypical with the severity mild. So the key concept is the genotype-phenotype correlation. If the phenotype is in line with the genetics, okay, there's no need to do second-level analysis such as sequencing or methylation. If the genotype-phenotype is suboptimal, we have to do additional tests. It is important to also to check when the genotype-phenotype correlation is not good. Also check also for a differential diagnosis, and also when possible to confirm the analysis by linkage analysis, and also following the analysis by segregation analysis. This is important because genetic disease also run in families. So I would like to spend some words about the genetic counseling. Is a communication process helping individuals, couples, and family to understand the disease, familial and reproductive implications, and so on. Even with the most advanced genetic and epigenetic tool, diagnosis alone is not enough. And patient and family need to understand what the diagnosis means, how it may affect their health, their relatives. So it is important to set importance on relatives. This is the first question they may ask, and their reproductive choices. So the genetic counseling, as I told you before, is a central role in bridging laboratory results in the clinical care. And so this is, in this cartoon, I like to show you the timing of genetic testing. Depending on the developmental stage at which the genetic testing is performed, we can distinguish between pre-implantation genetic testing performed before the implantation during the in vitro fertilization, typically on embryo created by in vitro fertilization. Second, we have the prenatal genetic diagnosis performed after conception, but on the embryo fetus, usually via chorionic villus semplus or amniocentesis. After that, we have the genetics in the adults when the clinical symptoms are present. We have two different kind of genetic testing: the direct and indirect. The direct is when we can analyze directly the causative mutation. Indirect is when we cannot analyze the causative mutation, but we can follow the segregation of chromosome harboring the disease. It's important to understand that, because so in this slide I want just to give you a schematic representation of the difference between direct and indirect. It is important to outline that sometimes the huge amount of material that is necessary for us to perform a Southern blotting cannot be feasible, for example, for pre-implantation genetic testing for the form 1. And so we have to follow the chromosome within the family. If the parents are present. So there's the need for a specific setting up. It is important to outline that the question is the implantation of prenatal testing feasible in facioscapulohumeral dystrophy. The answer is yes, but it depends. We have to understand first if we have a monogenic, so only the contraction. We have contraction and mutations, only mutations. If we have epigenetic modulator, we have to understand within the family the correlation, the genotype-phenotype correlation. We have to understand if we are in case of familial or sporadic cases. Sporadic cases makes some pre-implantation genetic testing, for example, really challenging, and in some cases impossible. We have also to plan in a deeper way the molecular analysis to perform before testing. What about the pre-implantation? There are 5 steps. The first 2 is the setting up. We have at the beginning, we have to understand if we can apply the pre-implantation genetic testing in the patients. We need time to do that, to plan the analysis. And after that, we have to perform the examination of embryos. What about the prenatal testing? It is performed after conception, and it allows the couple to obtain diagnostic information during pregnancy. It is important to outline that it is difficult to have a clear prediction of the phenotype, as I told you before, and therefore starting only based only on genetics is not so easy to be precise enough to predict the phenotype. And so, for these reasons, the prenatal diagnosis should only be offered when a well-characterized pathogenic allele is present in the family, when it is characterized by early onset of severe facioscapulohumeral dystrophy, and is also essential to perform a pretest genetic counseling to explore expectation of patients and ethical consideration. So these are in this slide, I want to summarize all these things about the prenatal testing and pre-implantation genetic testing. So it is important to keep in mind. I'm going to end just 2 or 3 minutes. So in this slide, I want to illustrate the optimal scenario that could be. So when we have test time for the genetic counseling, the setup and genetic counseling, the analysis of the first trimester, the CVS sampling, for example. After that, the post-genetic counseling. So for prenatal testing or pre-implantation genetic testing, but for prenatal testing, we need time. Patients need time to understand exactly what does it mean, but healthcare providers need time to set up the analysis. Unfortunately, the real situation is that many couples approach the genetic service only after pregnancy, and therefore they request prenatal diagnosis during the first trimester. This late time creates pressure for us as laboratories, but also for the counseling team, as the time required to set up the necessary assay is often really compromised. So, in my opinion, it underscores the need to promote early awareness of the genetic counseling, especially in family with a known history, positive history of the disease. Okay. So online, there's the presence of some information that are important for patients to understand all these things. And finally, I would like to show you a case important to summarize in a practical way what we have done so far. So this is a very illustrative case. The proband, the 2 proband, the number 2, received a diagnosis at age 30 with a severe phenotype. But in contrast, the mother is affected, but with a very, very, very mild phenotype. This is a classical situation of intrafamilial clinical variability between patients. So when we perform the analysis of the contraction, we observe the same contraction for repetition for both the mother and child. After that, we decided to look forward to the methylation analysis and whole exome sequencing. What about the methylation? The methylation revealed some differences between the mother and son in line with the phenotype. The proband had a much more severely hypomethylated profile respect to the mother. And so we decided to perform a whole exome sequencing. And what we discovered was that there was a pathogenetic variant in the LRIF1 inherited from the mother, and a second variant in the SHMID one coming from the father that was clinically unaffected. So the co-occurrence of the 2 mutations perfectly explained the more severe phenotype in the child respect to the mother. And it also highlighted the importance of testing both affected and unaffected parents as modified mutation may be inherited in a recessive mode of dominant mode, also from patients that are unaffected. What are the take-home messages? The facioscapulohumeral dystrophy is genetically and clinically complex with a spectrum of molecular mechanisms converging on the DAX4 gene. The traditional techniques like Southern blotting remain essential, the gold standard, but they must be now be integrated with modern technologies. That the new guidelines now follow a target structure where the level of genetic and epigenetic analysis is adjusted based on how well the clinical findings correlate with the molecular data. The DNA methylation is a powerful functional biomarker that can be useful for the diagnosis, patient stratification, and research. That the genetic counseling is fundamental part of the diagnostic process, that we have to follow a multidisciplinary, multimodal approach, combining clinical expertise, molecular genetics, and genetics to perform a very updated genetic diagnosis. And the last message for patients is that research progresses every single day. I would like to thank also all my collaborators in Rome, and I would be happy to take your question. Thank you.
Thank you very much, Emiliano. I forgot to say that FSHD Europe has also been involved in this presentation.
Yeah.
Nicole has an important leadership role in that organization, and so I apologize because I should have said it from the beginning. I cannot see questions, but Eugenio Zapata has his hand raised, so I'm going to unmute him. Eugenio, please unmute yourself and put your question.
Hi, can you hear me?
Yes.
Yes.
Hi. Thank you for the lovely presentation. It was very interesting. It helped me to clear some questions that I have. I wonder if I can ask you about a case that I recently had. If you have time, or if I can send you an email or something. But I had the case of a child that was sent to the genetic clinic because she had some motor delay since the first months of age. So they started doing the clinical workup and all the genetic workup, and they did a whole exome sequencing, finding a pathogenic variant in SEMCHD1. So when I saw her, I didn't think that it could be FSHD2 because she was a child, and I have in my mind that FSHD1 has a clinical presentation in children, and FSHD2 is in adolescents and adults. But she had some mild weakness. She had some mild facial weakness, a mild proximal weakness, but otherwise she's doing well. She's a very smart child, but she has a lot of fatigue. So I thought it could be a myopathy, and we did all the work for thinking for to correlate the SMCHD1. It was a de novo mutation, and then she had, we did the methylation study, and she had a DOX4 deletion. We can say that it's an FSHD type 2 and a typical severe FSHD type 2, but I'm still not 100% convinced it is FSHD type 2. And in the lab, they told me about doing other workup, but I don't know. Have you seen patients so young with FSHD type 2 with an insidious phenotype?
Okay, so in our thank you. First of all, thank you for your question. No, in my experience, as you have mentioned, on average, the facioscapulohumeral dystrophy type 2 patients have a later onset. However, I would like to to give you just some of my thoughts about that. So the first thing is that we have to understand these patients within the family. So how it's important to understand if this genetic characterization, methylation analysis, Wool exon sequencing has been performed also on the parents or not, if there are other sons or not. Yes. The second one is that did you find a mutation in SMID1 or other modifier genes?
Other modified genes? No, I couldn't. I don't believe that we have, because it was done through whole exome sequencing. So we only found the SMCHD1, and then we performed the methylation for the D4Z4 region.
Yeah, but I'm sorry, probably I missed some information. The patient is positive or negative for the SMID1 mutations?
Positive.
Okay, so
the
genetics
and the parents. Okay, and the parents are not affected. The parents.
Okay.
It's a de novo mutation.
So the genetics doesn't lie. So if we have a mutation, if we have a permissive allele, if we have a methylation, the diagnosis, in my opinion, could be done. But in any case, so to leave some space also to others. So I will be really happy to have the documentation. You can write me an email, and me and my team will be really happy to work with you on this case.
Well, thank you very much. It will be very helpful.
Thank you.
Got in touch to pursue a little bit more, but thank you, because I really want to be 100% sure that this is it.
Yeah.
Thank you.
Okay, that was useful. Now, let's go to the next question that comes from Raza Taraber, because I think it's very meaningful. So after what you said about all the requirements to have a good setup for a genetic lab and the guidelines to define exactly the conditions, the difficulty of common clinicians that may not be FSHD dedicated ones is to find an adequate lab either on their country or abroad. So is there anything that can guide us in that pursuit, in that investigation of where to send your samples?
Yeah, so this is a very interesting question, very important. If you note, one of the main time-consuming effort we have done in the preparation of the international guidelines of the update was the update also of the list of the centers providing the genetic diagnosis for 1, for 2 methylation Wool exon sequencing. All this information now are present online. I do believe that also the new instruments such as the chatbot based on artificial intelligence can provide patients with this kind of information, and the importance of the patients' associations cannot be overestimated. So to help you to understand, in my laboratories, the 75% of researchers are paid by a patient's association. Without this kind of association, all this work would have been impossible. And so we have to combine the efforts to put together our forces to make it possible.
Very good. When you say that that list is available online, you mean on the FSHD Europe website?
Yes, it is present. It is present in the paper in itself, but also directly on the website.
Okay.
The link is in the chat.
Thank you, Nicole.
Good.
So there is another question that is from an anonymous attendee that he or she would like you to develop a bit the idea of FSHD being a group of diseases instead of a disease. Why do you say that, and how do you distinguish between the four?
Yes, no, it's important because I probably I was not clear in that piece of my talk. So at the beginning of the story, we had 2 different diseases: the form 1 based on the contraction of the allele, and the form 2 based on the presence of mutations in the modifier genes, together with a borderline contraction of the allele. So these two disorders are not distinct disorder, but it is the same disease that can be present in form 1 with prevalence or form 2 with prevalence, depending on the combination of the genetic analysis. So it is important to outline that the facioscapulohumeral dystrophy form 1 and 2 share exactly the same features. So, we can have some slight differences, but it is important to say that we have a single disease that can be caused by a combination of different genetic and epigenetic factors, and that in the view of this combination, we can have a different expressivity of the disease. In my last example, I show you exactly what this kind of different level of genetic testing can be important. For example, probably 5 years ago, we concluded that the family had an intrafamilial variability for unexplained genetic factor. Now, this variability can be explained. The disease is just one, but the clinical expressivity can be different in patients because the mutations can be different. And it is important to outline that some mutations can be inherited from the healthy parents. It is important to understand, especially for prenatal testing and pre-implantation genetic testing. It's crucial, in my opinion.
That's good. I think that we need to come back to this theme. This sounds a bit like the quantum mechanics of genetics. It's too complex to absorb in a webinar, but I think that if we re-inject people with this information over and over again, we'll eventually have the possibility of understanding a bit better the complexity and then manage better our patients. I don't know if Nicole wants to close with some words of wisdom.
Well, I would like to thank Emiliano for providing a very nice overview of clinical testing from a medical genetics perspective, which was very useful. I want to add that the reason why I initiated the development of the recommendation was also because I had difficulties in the recruitment of clinical trials. And for clinical trials, patients need to be tested according to current standards, and now we have these current standards, and I think that is very helpful. So it was very helpful for myself to be involved in this project because it's supporting the recruitment in future trials. And finally, I would like to say thank you to Antonio and to Ria de Haas, whose name was mentioned on the last slide, and she's the contact person for FSHD Europe. And maybe you can, well, we don't have to show the slide. If you have questions about FSHD Europe, Emiliano can forward you to Ria de Haas.
Yeah.
Excellent. So I want to thank Emiliano, Nicole, Ria de Haas, and all the attendants for this very nice opportunity to learn and a very nice webinar for our collection. Please, when leaving, please click the link on the chat to give us feedback if you are all right with that and you enjoyed it. And next week we'll have another webinar about FSHD. As I said, it's the month where we have dedicated webinars to this thematic. So thank you everybody and goodbye.
And see you next week. Bye.
Bye.